hear me, Mom? So we are in group 5 and we are assigned to tackle about the different metabolic pathways. So as we all know, we have four different pathways, and we have carbohydrates, lipids, proteins, and nucleic acid. So these four I discussed namin. So I am assigned for the carbohydrates. But before that, what is metabolic pathways? So as we all know that metabolic pathways is integrated networks of biochemical reactions that occurs within cells responsible for converting molecules into, we have energy, signaling molecules, and also building blocks. So, carbohydrates na po tayo. So, carbohydrates, metabolism, refers to the biochemical processes that involve in the breakdown synthesis. Utilization of carbohydrates by living organisms. So, carbohydrates are organic molecules composed of, first we have carbon, hydrogen, and oxygen. So, basic carbohydrates, it might be mono, which means saccharide, which means one, disaccharide, and disaccharide, which means two, and poly, which means more than two. So, monosaccharides, simple sugars and glucose, fructose, and disaccharides, double sugar forms that formed by the oxidation of two monosaccharides. So, we have example like sucrose and lactose. And lastly, for polysaccharides, it is complex. As we say that more than two, it is a complex of carbohydrate consisting of long chain polysaccharides units of glycogen, starch, and cellulose. So, under carbohydrates metabolisms, 5 types of carbohydrates. These are the 5 types of carbohydrates. So, they have glycolysis, gluconeogenesis, glycogenesis, glycolysis, and lactic acid cycle or PCA cycle. So, what is glycolysis? So, glucolysis is a carbohydrate metabolic pathway that converts glucose into pyruvate. Glucose into pyruvate. Located or occurs in cytoplasm. That is according to the law. So, they break down glucose into pyruvate and producing ATP and NADH. Next this glycolysis, we have the phosphorylation, cleavage, oxidation, and ATP generation. So we have products is 2 molecules of pyruvate and 2 molecules of ATP and 2 molecules of NADH. So ito na yan. And then yung structural type niya, and then may your hope pathway. So key factors niya is converts glucose, a 6 carbon molecules into 2 molecules of pyruvate. Pyruvate is a 3 carbon molecules and then 10 enzymic steps in the cytoplasm generates in ATP and NADH. So ganito siya. So in glycolysis, we have 3 stages. So yung first stage is invest ATP, second is yung 6 carbons split into 3 carbons, and third is yielding base. So glycolysis, we have 3 stages of glycolysis. So first stage is we have naming building ng glycolysis in the first stage, tapos ito yung parang structure niya. So, glucose, we have ATP, ADP, or we call is also called as hexokinases, or hexokinase. So, papunta ay next is glucose 6-phosphate, which is phosphoglucose isomerase. Then fructose 6-phosphate, phosphofructokinase to ATP, ADP. And that is fructose 1,6-biphosphate. So as you can see, bracket bi, mostly we use di. Pero same na sila kasi we have two number of atoms na naka-attach si phosphate. So, dito tayo sa structural, as we all know that glucose has 6 carbons. So ganito yung structure ng glucose, daanan natin yan. And then glucose 6-phosphate, as you heard 6, so yung phosphate is naka-attach to number 6. So PO3 is the phosphate. Next is fructose 6-phosphate. So fructose 6-phosphate, as we all know that fructose has 5 carbons. Bakit 6? Because the phosphate is attached at the 6 carbons of the fructose. So 1, 2, 3, 4, 5, 6. So in 6 carbons sa fructose, doon naka-attach si phosphate. And last is fructose 1,6-biphosphate. Ito yung sinasabi natin, dalawang phosphate na naka-attach. As you can see, we have from 1, 2, 3, 4, 5, 6. And 6. So fructose 1,6-biphosphate. So combinations of stage 1 and stage 2. So in stage 2, the 6-carbon sugar is split into 2 carbons. So, dito po siya. So, in stage 2, we have aldolase. In aldolase, we have yung triphosphate isomerase split into 2. We have glyceraldehyde, 3-phosphate, and hydroxyacetone phosphate. So, in acetone phosphate, ito yung so, same attach of the potato with CH2 of OH and CH2 of the presence of phosphate or OP3 to negative. What is structure niya? We have 3 carbon. Attached is OH or presence of OH, then CH2 OPO or phosphate 3 to negative. So next we have stage 3. So stage 3 is an energy yielding phase. The oxidation of 3 carbon fragments yields ATP. Stage 3, this is the condition of stage 3, which it identifies how the stage 3 flows. So in the first step, it is given as 1/3 by polyglycerate, which is structured is this one. Bakit why? Because in first carbon is attachment of the phosphate or glycosylate. Ito po, and then in the third carbon also is attached here, glycosylate. So, glyceraldehyde, 3-phosphate dehydrogenase, or also called as 1,3-bipoglycerate, which is in this two. Nasa dito po, dito siya si PI-NAD, positive, and NADH. Second, we have the 3-phosphoglycerate, which is ito po. Bakit 3 na lang? Because the phosphoglycerate is only attached to the third carbon. So, ganito po siya. So, to the next. So, in the 3, we call it as phosphoglycerate kinase. So, ang presence dito is ADP and ATP. So, in the next, we have 2-phosphoglycerate, which is, ang presence dito is phosphoglycerate butase, which naman, ganito po yung structures niya, which is, we have 3 carbon. In the first carbon, there are 2 attached of oxidate. O ganon. Tapos, second, we have the phosphate or phosphoglycerate. So, the third carbon is another attachment of OH. And then the third, we have phosphoenolpyruvate. Kasi dito naka-attach, dito na si H2O. And then we call it as enolase. And sa structures naman, nakikita natin, hindi siya actually na double bond, pero ito yung structure ng phosphoenolpyruvate. And the last is pyruvate. So in pyruvate, we have isang daan na lang, which is naandun yung presence ng ADP and ATP, which we call pyruvate kinase. So in the structures naman, ganito po yung structures ng pyruvate. Next we have the example of phosphoglucose isomerase. So as you can see, we have the phosphoglucose isomerase, the conversion of aldose to ketose. So we have the earlier kanina glucose to 6 and glucose is phosphate. Now carbons is naka-attach yung phosphate. So 1, 2, 3, 4, 5, 6. Ngayon, phosphate. And then, Mg2 positive, phosphohenose isomerase to fructose 6-phosphate. Bakit nga ba natawag natin fructose? 6-phosphate, since the fructose naman is 5 carbon B. So, let us count. 1, 2, 3, 4, 5, 6. So, in the 6 carbons, dito naka-attach si phosphate. So, this is an example of phosphoglucose isomerase. So, this is the isolation of phosphoglucose isomerase. So, as you can see, the enzyme opens the rings, catalyzes the isomerations, and promotes the closure of 5 member of ring. So, di ba kanina, ito pala yung gawa. So, the end is here. So, now, makikita natin dito yung, what do you call this? Fisher form. So, I'm sorry. Fructose 6.
So that is a glucose and the fructose.
So, glucose 6-phosphate. Ito yung Fisher form na yung 6 carbon naka-attach si phosphate. 6-phosphate. Kaya siya natawag na 6-phosphate. So, in the fructose naman, we have 5 number of carbons and number 5 to 6 yung attach yung phosphate. So, ay, dito 6. 1, 2, 3, 4, 5, 6. So, ganito po siya. F, 6B. Mg. Ito yung isomerase of phosphoglucose. This is one called phosphorylation transfer. So, we have an example of phosphorylation transfer reaction. So, as you can see, kinesis transfer phosphate from ATP acceptor. So, hexokinesis has more general specific in that it can transfer phosphate to another sugars as more malose. So, for example, we have the glucose. Glucose transferred hexokinase to glucose 6-phosphate. So, alam naman natin yung yung structure of glucose. We have 1, 2, 3, 4, 5, 6. So, in 6, ganito siya. And then, we have here the kinosase, Mg2 positive, ATP, and ATP. So, here, bakit siya natawag glucose? Since may glucose tayo dito presence of glucose, 6-phosphate because the phosphate is attached at the 6 carbon. So, that is an example of Rosebery transfer reaction. Phosphofructokinase or PFK. So, this is the structure of phosphofructokinase with fructose and example of fructose 6-phosphate. As we know that fructose has 6 carbon and the presence of phosphates, and on the other hand we have the fructose 1,6-biphosphate. So why is bi? So mostly kasi, di ba, bi use di. So same yan sila. Bi is two kinds of 2 carbon that presence of phosphate. So 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. So, in the 6 carbon, andito po nakatouch si phosphate. So, on the other hand naman, so in 1 carbon attached si phosphate and 6 carbon attached si phosphate. So, phosphofructokinase. So, ang ATP, if may presence of magnesium, is magiging siyang ADP. So, PFK, which means it is an important allosteric enzymes regulating the rate of glucose catabolisms that plays roles in integrative metabolisms. So next we have the aldolase. So in aldolase we have fructose 1,6-biphosphate and the presence and aldolase 2-dihydroxyacetone phosphate and glyceraldehyde phosphate 3-phosphate. So as usual, fructose have 5 carbons. So attach the phosphate in 1,6. And then, so aldolase, we have dihydroxyacetone phosphate, which is ganito. We have 3 carbons. In the first carbon, dito yung attached is phosphate, and the third carbon attached is the presence of OH. Plus, the glyceraldehyde, 3-phosphate, which is same is 1, 2, 3 carbons. Pero, in the second carbon, attached dito si OH. First carbon is ganito, presence of O and H. As with double bond. So, in the third carbon is naka-attach na dito si phosphate. So, kaya 3-phosphate kasi si carbon is in 3-phosphate. So, next we have the triose-phosphate isomerase. So, in triose-phosphate isomerase, all the DHAP is converted to glyceraldehyde 3-phosphate, although the reaction of the reversible is shifted to the right since glyceraldehyde 3-phosphate is a substrate for the next reactions of glycolysis. Thus, the both three carbon fragments are subsequently iodized. So, structures ng dihydroxyetone phosphate and glyceraldehyde phosphate. So next we have gluconeogenesis. So gluconeogenesis is a synthesis of glucose from non-carbohydrate precursors. So glucose are stored during the periods of starvations or fasting. So since the brain relies on glucose of 120 grams per day as the source of the energy, glucose must be synthesized from the molecules other than carbohydrates. So this is what we call pyruvate to glucose. Glucogenesis from pyruvate to glucose, so any molecule that can be converted to pyruvate is considered as glucogenic. So lactate and alanine, also glycerol, is considered to glucogenic. So this is the structure of glucogenesis. So we have glycerol kinase, glycerol phosphate to NAD positive, then to NADH, presence of hydrogen positive to dihydroxyetone phosphate. Glucogenesis pyruvate enzymes in red belong to gluconeogenic pathway. These reactions overcome the high negative free energy from irreversible reactions of glycolysis. So the enzymes in blue are held in the common between of two pathways. Gluconeogenesis, the enzymes of glucogenesis are ATP or pyruvate carboxylase, and also GTP or what we call is phosphoenol pyruvate carboxygenase and fructose 1,6 biphosphatase and glucose 6-phosphatase.
So glycolysis, to make it short, it is located at cytoplasms, which means glycolysis means that involves breakdowns of one molecule of glucose into two molecules of pyruvates, which actually the glycolysis doesn't require oxygen in the first step, so it also generates a small amount of ATP and NADH. So we have the 10 steps of glycolysis. First, we have the hexokinase. Hexokinase is where glucose is phosphorylated by ATP to form the glucose 6-phosphate, catalyzed by the enzymes hexokinase. So second, we have the phosphoglucose isomerase. So when you say phosphoglucose isomerase, it is a glucose of 6-phosphate is converted to fructose 6-phosphate by an isomerase enzymes. So next we have the phosphofructokinase. When you say phosphofructokinase, it is a fructose of 6-phosphate, is further phosphorylated by ATP to form a fructose 1,6 biphosphate phosphofructokinase 1 or PFK1. So next we have the stage 4 is allulose. When you say allulose naman, it is a fructose of 1,6 biphosphate is split into 2. So it's split into 2, then 3 carbon molecules hydroacetone phosphate or DHAP and glyceraldehyde by 3-phosphate or G3P. So next we have the steps 5, which is triphosphate isomerase. In stage 5 naman, we have triphosphate isomerase. DHAP is converted into another molecule of G3P. So now there are 2 molecules of G3P. So in stage 6 naman, we have glyceraldehyde 3-phosphate dehydrogenase. So when you say dehydrogenase, step 6 glyceraldehyde 3-phosphate dehydrogenase. It is each G3P Each D3 molecule is oxidized and phosphorylated, result in the production of 2 NADH and 2,1,3 by phosphoglycerate molecules. So in step 6, we have the glyceraldehyde 3-phosphate dehydrogenase. We say step 6, each of the G3P molecule is iodized and phosphorylated, resulting in the production of 2 NADH molecules of 1,3 biphosphorylated molecules. So in step 7, phosphoglycerate kinase. Ito naman is in phosphoglycerate kinase, 1,3 biphospoglycerate is converted into 3 phosphoglycerate with the concomitant generation of ATP through the substrate's level of phosphoglyceration. So next, we have 8th step, the phosphoglyceromatase. The phosphoglyceromatase, it is a 3 phosphoglycerate is isomerized to form 2 phosphoglycerate. So in step 9 naman, we have the enolase. Step 9, we have the enolase. So enolase, it is naman 2 phosphoglycerate loses water to become phosphonol pyruvate or PEP. So in 10 step, we have the pyruvate kinase. This is the last step, which the PEP is converted into pyruvate while generating ATP through the substrate level phosphorylation. So in summary of this glycolysis consists of 10 enzyme steps. So 10 enzyme relations that convert one molecule to glucose into 2 molecules of pyruvate, producing ATP and NADH along the way. So next we have after glycolysis we have the glycogenesis. So when you say glycogenesis, it is metabolic pathways that enables the body to synthesize glucose from non-carbohydrate sources, ensuring a steady supply of glucose for energy productions. So in glycogenesis we have 4 steps. So in first step we have the conversion of pyruvate to oxaloacetate, which means that it is a pyruvate carbohydrase converts pyruvate to oxaloacetate in mitochondria, a key step in increasing gluconeogenesis. So in step 2, we have the conversion of oxaloacetate to phosphonol pyruvate. So in step 2 we have the phosphonol pyruvate carbocyclinase converts oxaloacetate to phosphonol pyruvate in the cytoplasm, providing a precursor of glucose synthesis. So in stage 3, the conversion of fructose to 1,6 biphosphate to fructose 6-phosphate. So in step 3, we have that conversion of fructose fructose 1,6 biphosphate removes a phosphate from group fructose from 1,6 biphosphate, leading to the formation of fructose 6-phosphate. So number 4, we have the steps 4, we have the conversion of glucose 6-phosphate to glucose. So in step 4 naman, we have the conversion of glucose 6-phosphate to glucose 6-phosphate catalyze the final steps in gluconeogenesis by the converting glucose 6-phosphate to free glucose, which can be released into the bloodstream. So to make a recap of it, so glyconeogenesis, it is a vital metabolic pathway that allows the body to generate glucose from non-carbohydrate sources to maintain blood glucose levels and provide energy to tissues during periods of fasting and low glucose availability. By converting the substrates through a series of enzymatic reactions, glycogenesis ensures that a constant supply of glucose is essential for psychological functions. Next, we have the glycogenesis. So After the glycogenesis, we have the glycogenesis. Glycogenesis is a metabolic process in which the glucose molecules are converted into glycogen for the storage in the liver and muscle servings as a reserve energy source. So in glycogenesis, we have also 4 steps. We have first the formation of glucose in 6-phosphate. Second is the formation of UDP. Glucose. Third is glycogen chain elongation. And fourth is branching of glycogen. When you say first formation, the first step is formation of glucose in 6-phosphate. It is before that we have the short functions of the glycogenesis. We have the primary functions of glycogenesis is to store the excess glucose as a glycogen when blood glucose levels are high, providing a steadily available source of energy for the body when needed. So formation number 1, formation of glucose 6-phosphate. Glucose is phosphorylated to the glucose 6-phosphate by hexokinase, chopping it inside the cell and preparing for the glycogen synthesis. So in the second formation of UDP glucose, it is also glucose of 6-phosphate is converted into UDP. Glucose by UDP glucose pyrophosphorylase activate glucose for the incorporation into the glycogen. So third, we have the glycogen chain elongation. When you say glycogen chain elongation, the glycogen synthase adds UDP glucose units to the growing glycogen chain, extending it and formation alpha 1,4 glycosidic bond. So, in 4 steps of the glycogenesis, you have the branching of glycogen. When you say branching of glycogen, it is the branching enzymes introduce alpha 1,6 glycosidic bonds to create the branch structures within the glycogen molecules, increasing its solubility and accessibility for rapid energy release. So for a short recap, glycogenesis is a crucial metabolic pathway that converts glucose into glycogen for a storage, ensuring reserve energy source for the body. So through this, through a series of enzymatic reactions, glucose is activated, added to the growing glycogen chain and branches to enhance its efficiency as everyone serves. So glycolysis importance, it plays a vital role in regulating the blood glucose levels and providing a quick source of energy during the periods of a fasting or increasing of energy demand. So it allows the body to store access glucose efficiently in the form of glycogen, ensuring the stable energy supplies and support of metabolic homeostasis. Additionally, glycogen serves as a critical fuel source of muscles during exercise and helps to maintain glucose levels in bloodstreams to support the brain functions every balance. So next we have the glycogenolysis. So glycogenolysis, it is a metabolic process where the glycogen stored in the liver and muscles is break down into glucose to provide a rapid source of energy for the body. So when you say glycogenolysis, it is a primary functions of glycogenolysis is to mobilize glucose from glycogen stores during times of low blood glucose levels or increased energy demand, such as during the exercise or fasting. So we have 4 steps for the glycogenolysis. So first we have glycogen phosphorylase. Second we have the branch point cleaving transitions of glucose to phosphate. Fourth is final conversion to free glucose. The first step is glycogen phosphorylase.
So we say glycogen phosphorylase activation is the glycogen phosphorylase is activated by the hormone glucagon or adrenaline initiating the breakdown of glycogen into glucose 1-phosphate. So, second step is we have branch point cleavage. So, when you say branch point cleavage, it is that the branching enzyme removes branches from the glycogen molecules, allowing glycogen phosphorylase to continue break down the linear chain into glucose units. So, third, we have the conversions of glucose phosphate. When you say conversions of glucose 6-phosphate, phosphoglucomatase converts glucose 1-phosphate to glucose 6-phosphate, a critical intermediate in glucose metabolism. So fourth step, we have the final conversion of free glucose. When you say final conversion of free glucose, it is the glucose 6-phosphatase further processes glucose 6-phosphate into the free glucose, which can be increased into the bloodstream for the energy production of various tissues. So, for a short recap, we have the glycogenolysis. Glycogenolysis is a metabolic pathway that breaks down glycogen into glucose for immediate energy needs. So, through a series of enzymatic reactions, glycogen is rapidly converted into glucose, providing a quick, efficient energy source from the body. So, its importance is glycogenolysis is crucial for maintaining blood glucose levels within a narrow range, especially during fasting, exercise, and times of stress. So, by releasing glucose from glycogen stores, glycogenolysis ensures a constant supply of energy to tissues like the brain and muscles, supporting overall metabolic functions and physical performance. So, additionally, glycogenolysis helps prevent hypoglycemia and provides a rapid response to energy demands, contributing to the body's adaptability and resilience in the various psychological conditions. So next, we have lastly the critic cycle or Krebs cycle, also known as critic cycle. So it is a series of chemical reactions that occurs in the mitochondria of the cells to generate energy through the oxidations of the acetyl CoA. So we have critic cycle or Krebs cycle. We have 8 steps. So first we have the acetyl CoA formation. Second, the citrate isomerization. Third, first oxidation. Fourth, second oxidation. Fifth, substrate level phosphorylation. Sixth is third oxidation. Seven is hydration. Eight is fourth oxidation. So when you say acetyl CoA formation, it is acetyl CoA derived from the breakdown of glucose fatty acids and or amino acids. So enters the cycle by combining with the accelerated oxaloacetate to form. Second, we have the citrate isomerization. So when you say citrate isomerization, it is a citrate undergoes the isomerization to form an isotrate. So third, we have the first oxidation. When you say first oxidation naman, it is isocitrate is iodized, leading to the release of carbon dioxide or CO2 and the formation of alpha ketoglutarate. So fourth, we have the second oxidation. When you say second oxidation, alpha ketoglutarate is oxidized, resulting in the release of another CO2 molecule and the formation of succinyl CoA and NADH also produced. So in fifth step, we have the substrate level phosphorylation. So it is phosphorylation. It is a succinyl COA is converted to succinate, and in the process, a molecule of GTP or ATP is generated. So since we have the third oxidation, we say the third oxidation, it is the succinate is oxidized to form fumarate producing FADH2 in the process. So 7, we have the hydration. When you say dry hydration naman is converted into malate through the addition of water. So lastly, the fourth oxidation. When you say fourth oxidation, it is a malate is oxidized to generate a oxaloacetate, generating another molecules of NADH in the process. So it's Krebs graph or a summary for Krebs cycle, acid cycle, or Krebs cycle. So the cycle then reacts as oxaloacetate is available to combine a acetyl CoA to begin the process again. So the overall outcome of the Krebs cycle is the production of the energy, which molecules in the form of NADH and FADH2 as well as some ATP and GTP through the substrate level phosphohydrolation, where their energy carriers go on to participate in the electron transport chain to generate ATP, which is the primary currency of